So if you don't have or need to print, right, this is label diagrams, it's under Module 13. The instructions for this part is match the following pictures with the outcomes. So which picture shows neutralization? So you could either put the letters to the pictures, A, B, C, D, or E on this slide if you want, or you could label these pages, or you could do both, right? Whatever helps you learn, right, each one of these 6 potential outcomes that can happen when antibodies bind antigen. Okay? That we'll go over on Friday. So today is the first part of Section 6, Chapter 20, which is the first diagram that you guys had to label for homework. Okay? So right here, they have written MHC class 1 or 2. So how many people, raise your hand, think it's 1? Okay? How many think it's 2? Obviously, the whole class is going to raise their hand now, right? That's pretty awesome because that's the right answer. Right? And really, what cell are they talking about that's doing that? Which one is expressing MHC class 2 that we just agreed upon?
T cell.
Who presents antigen? Dendritic cells, macrophages, one more, B cells. Which one at the top of this diagram do they say is presenting antigen? Okay, I will back up. Which one? Dendritic cell. Because you guys just said, I heard the whole class, you guys got it right. You said dendritic cells were antigen-presenting cells. You said macrophages are antigen-presenting cells, and you said B cells. At the top of this picture, you have a choice. An antigen-presenting dendritic cell or a B cell. Which one is expressing MHC class 2? I'm asking all of you. Dendritic cell. This one. So notice I put the label even over here next to this guy. Maybe I need a big screaming arrow right here. Maybe. And he is, yes, presenting it to, who is this? T cell. And it is a naive T cell. What does naive mean? It has not encountered antigen yet, right? And what they don't have drawn in here is that specific interaction between these two cells. Once this cell is activated, it's going to proliferate, right? So we have more of them to go out and do their jobs. So this is heading out to do their jobs, this branch. Some of them are going to be memory cells, right, that are going to stick around in case we see this guy again. So if an antigen-presenting cell, in this case dendritic cell or a macrophage, comes along later, it could present to this memory cell, and we start the process all over again. That's what this back arrow means, right? It means we go back and we produce more helper T cells and more memory cells even. So now we found out last time, right, there are 2 ways we could kind of go as far as the adaptive immune response. We can do a cellular response, which is this side, or we could do a humoral response, which leads to the production of antibodies, what we're going to focus more in on here today. So these helper T cells actually have been divided into 2 sets based on the cytokines they produce, and therefore what cells they talk to and what outcome we get. So in this case, right, for cell-mediated, talking to cytotoxic T cells, generating and activating cytotoxic T cells, or this lineage, the humoral, is helper T cells talking to B cells. So there's even a clue here I didn't cover up, but we'll get to that. So which subset is responsible for our cytotoxic cell-mediated response? TH1 or TH2? TH1. TH1.
I'm trying to watch this everybody paper at the same time.
It's okay. It's TH1. Okay. I didn't even make the class raise their hands. Okay, we'll do it for the next one. Okay, so then this cell is helping out this cytotoxic T cell, right? And we know it's cytotoxic because what does it have here? CD8, which means it can talk to this antigen-presenting cell, but this antigen-presenting cell must be presenting antigen via which MHC? 1. How many people say 1? All right. How many say 2? Anyone says 2? They changed their mind now because most people raise their hand for 1. 1 is correct, right? MHC class 1. And remember, our cells, they don't show this here, all present, for the most part, MHC class 1. Remember, this cellular response is for stuff getting inside of our cells. We want to be able to present that outward via MHC class 1 so that the CD8 cytotoxic T cells can recognize it and destroy it. And remember, whenever a T cell or B cell is activated, we're always going to produce memory cells. So if an antigen-presenting cell, whether it be a macrophage or a dendritic cell, sees this again and presents it to a memory T cell, cytotoxic one, we can start this process all over again, right? And much faster than trying to find a naive cell. So now on this other lineage, by default, right, which is it? 1? Anyone think it's 1? We already used 1, so what are we left with? 2, right? Everyone agree? Yes, right? So Th2 is our humoral, our B cell activation, and it says it, look, right here, Th2. So we already know from the helper T cell way up here, too, it gets presented antigen via MHC class 2, so guess what B cells do? MHC class 1? No takers? 2? What you think? How many people think 2? Raise your hand. High and proud, right? It's 2, right? It's 2. MHC class 2. And who is presenting? Right? Remember, you need to remember who is actually presenting is using the MHC class 2. It's picking up the proteins that it took in, right? And they're presenting them to the T cells. So this is a very good diagram. If you can take a blank form of this diagram and fill it in, you're golden. I should probably give you guys a blank one, completely blank, huh? See how you do on that. All right, so here it is with all the stuff filled in. So we're going to focus more on this, and we've talked a little bit about humoral, right? But we're going to focus in on humoral today. And you'll notice that there's just not one type of antibody that's produced. There's actually 5 different kinds. We're only going to focus on 4 of them, the 4 main functioning ones in this class. And we're going to talk about those next time. So recall, B cells produce antibodies, okay, in response to protein antigens from foreign sources, okay? So they internalize what binds to their B-cell receptor, and they're going to present that, right? Helper T cells are going to help them in that, as we just saw in this past diagram. The T-cell receptor actually specifically recognizes that antigen. It's going to stimulate those B cells to multiply, differentiate into plasma cells, which actually secrete antibodies, and memory cells. Again, whenever a B cell or T cell is activated, we're always going to get memory cells. So your first time you ever see an antigen, we've got to find naive helper T cells, right? We've got to find naive B cells that recognize this antigen. This could take a while, right? The dendritic cell is running around from lymph node to lymph node, right? Or within the lymph node in that area from where he picked up the stuff, and is trying to find somebody who recognizes what he's presenting so he can activate them. So that very first time, the unfortunate thing about that is it could take from 7 to 14 days to find a naive B cell, a naive helper T cells that recognize that specific antigen. All right, so this takes time. So who's going to save your butt and keep this under control in the meantime? What? Well, that's how long it takes to make an antibody. So you don't even have, this is the first time you've seen it. You've never seen it before. You haven't been vaccinated. You haven't gotten sick with this virus before. It's the very first time you've ever seen it, right? Or this bacteria, say, some foreign entity. You don't have any memory B cells, no memory T cells.
I'm guessing natural killer.
Yeah, natural killers to a certain extent, that's if it gets inside of your cell, right? Because natural killers kill your cells that get infected. Well, what if it's a bacteria and it's staying on the outside? Who's got to help you in the meantime until we start making antibodies to get this thing going a little bit more specifically? So what's the first thing that's probably going to happen if he invades into your skin? You hope. Right? You get a puncture, the bacteria get in, that's it. We just let them...
Is it the immune system?
It is blood component of your immune system, though. Your dendritic cells are going to pick it up, right? But they're not going to stay there and take care of the problem. They're going to run off to try and find help, right? That's their job, to present antigen. Who at the site is going to take care of the invasion? Macrophages. And then who else is he going to probably call in for reinforcements? That's really good eating cells as well. Because that's what the macrophages are going to do, right? They're going to phagocytize and destroy the invader.
Cytokine?
Cytokines are going to they're going to talk, right? The cells in that area are going to release cytokines. They're going to scream out for help. Neutrophils, right? But again, are they specifically recognizing that pathogen? Do they know that's E. coli 0157 that just got in? No. But they recognize general patterns, right? They recognize, oh, a gram-negative bacteria got in, right? I recognize it's LPS. I can bind to this. I can phagocytose it, right? I can destroy it. And they're going to keep eating stuff, but bacteria grow really fast, right? And they can only eat so many. So if they can't get it under control, right, dendritic cells already run away to go find help, right? But this help could take 7 to 14 days. This is why the fact that we produce memory cells is so awesome. Because we take advantage of this by vaccinating people, right? We give them a weakened form of the pathogen and activate naive helper T cells, maybe even cytotoxic T cells, B cells, and they produce memory cells. So that when we get invaded again, we don't have to wait that 7 to 14 days, right? We've got more cells out there, memory cells, that the dendritic cell and even the macrophages can present to and activate. So that's referred to as our secondary, or more often now we refer to it as our memory response. Because again, it's the memory cells in that case that are being activated. And the response is much quicker because of that. So say antigen A, right? The very first time you're exposed, it could take up to 14 days before, when we test your blood, we see substantial levels of antibodies in your system. The second time you're exposed, memory cells. So notice, see how quick the response is? And even much more vigorous than the first response. Right? We're not messing around the second time. They say, fool me once, shame on me. Fool me twice, shame on me. Shame on you the first time, right? Shame on them the second, yourself the second time for not knowing better, right? The second time around, you should know better. So your system knows better the second time around. It's like, oh, I've seen this before. I ain't messing around. Notice that you could have you're having a primary response right here. You have a secondary or memory response because you've seen antigen A before. But let's say antigen B comes along with an injection of antigen A again, right? A, it's a secondary response, a memory response, because you've seen that one before. But B, it's the first time you've seen it. So you're going to have that primary response, which again is going to be delayed. Because you've got to find naive cells that recognize. So again, it's antigen specific, right? Have you seen it before? If you have, you're more apt to respond much faster and much more aggressively. So as we talked about, B cells have a B-cell receptor. It's actually their antibody on their surface. So unlike the T-cell receptor that only interacts with one little piece of the antigen, the B-cell receptor can actually interact with two pieces of the antigen or even two separate antigens. And we'll look more closely at that, hopefully, if we have time today. Okay, so it's going to internalize this, what's binding to its antibody. But in this case, notice it's a lipid. They're saying it's a lipid or a carbohydrate. But what have we been referring to antigens for the most part as? Proteins. Because that's what MHC can present, is proteins. It can't pick up anything but a protein. So we have a problem here, right? We've got, it's not a protein. It's a sugar or a fat, a lipid, that's being internalized. But when these B cells internalize stuff, they pick up proteins in the area. So they might be picking up other pieces of the bacteria or the whole bacteria itself, or the virus. And they're going to process that, and it's the protein components that are actually going to get presented via MHC class 2, right, to the helper T cell. We know this is the subset 2, the Th2 subset, that is going to help stimulate these B cells. So the B cells will be stimulated, they'll proliferate, some of them will turn into plasma cells that actually secrete the antibodies. Others turn into memory cells, right? That antibody binds to what it originally bound to. In this case, they're saying the capsule that's made out of a polysaccharide out of a sugar for this particular example. But they for that example and for what we've talked about so far to this point, right, you need T cells and therefore we need some protein that's antigenic to be presented. So they take this into consideration when they're developing vaccines too. There's always a protein component or even a component that'll bind to toll-like receptors on macrophages and dendritic cells, so they will take it in and present it. Okay, but our B cells may bind to different portions of that may not be protein. They could be sugars, right? They could be fats. But we need that protein component in order to get this level of activation. And you want this level because this is the level that's going to give you memory cells. So as is shown in the diagram, the B-cell receptor, right, that antibody in its surface, may take up non-protein antigens, so long as antigenic fragments can be presented on MHC class 2. It can solicit T cell help, right, and drive that antibody response. So in this way, even though the antibody is responding to, say, a carbohydrate or lipid, it can still be activated. We can still get that antibody, that humoral immunity response. Well, when they were studying mice, they found that, and even for some humans that don't contain a thymus, because remember, the thymus is what the T cells are named after, because that's where they mature, right, and become helper T cells and cytotoxic T cells and know the difference between self and non-self. So if you don't have T cells to activate B cells, how is it that these humans or these mice are still producing antibodies? There must be an exception to the rule, right? And in fact, there is, right? There are some antigens, the way they interact with the B-cell receptor, the antibodies on the surface of the B-cell, they can activate it, but not fully activate the B-cell, right? So these are termed T-independent antigens, because as we just said, if you don't have any T cells, there's no way T cells are helping out, right? It's independent of T cells. So there are some antigens out there are called T-independent. You don't need B-cell help in order to produce antibodies. So the next question is, how do they stimulate it then if we're not having B cells interact with T cells, right? How does this happen? So typically, these types of antigens, these T-independent antigens, are long polymers. What's a polymer? Anyone know? Poly means many, right? MERS referring to the subunit. So a protein is actually a polymer, right? It's made up of a lot of different amino acids. Molecules like LPS, right, that have a lot of repetitive components to them. And they're really long molecules, right? Polymers are big molecules, right? Poly meaning many components to them. They're so big that they can interact with more than one B-cell receptor on the surface of the B-cell. So we'll look at that in a moment. That triggers the B-cell to proliferate, produce plasma cells, but they stay at the base level of antibody production, which is the first antibody that we produce, and which is the one that's on the surface of your B-cell, is called IgM. But when you secrete this antibody, this one I always remember M for mega big, right? Because this guy is huge. It is 5 antibody subunits hooked together to form this huge complex. So these types of antibodies have lower affinity. Anyone know what they mean when they say affinity? So antibodies bind to antigens, right? They don't bind that strong, right? That's what affinity means. How strong that binding is, right? How much do they interact and stay stuck together, right? So low affinity, does that mean they stick together really well? No, it means it's not a very, but it's a close enough match, right? It's not like a fingerprint, though. It's not perfect, right? If you're using your fingerprint for your phone, right, it's not going to work, right? Something's happened. It's off. It isn't close enough, right? But for other things, it would work. So, and so this is a weaker response, often not, but not always, but often not even producing memory B cells, right? So we're not so sure about it, right? We don't want to be too aggressive, and we might not even need to remember it. Typically, less antibodies are even produced than when we have that stimulation from T cells. So this is what it looks like. So this is where, as you can see, a lot of the molecule is interacting with a lot of the B-cell receptors. That's enough to activate that B-cell to proliferate, produce plasma cells that secrete only this class of antibody, the very first one we produce, which is IgM. We can switch to other classes that we'll talk about next time, but you can't do that unless the B-cell is helped out by the T-cell, right? You can't make that switch to something else if you don't get communication from the T-cell. So as I said, these guys are really big, right? So now we're gonna zoom in on antibodies and talk about the different components and how they work of this protein. So plasma cells can secrete 2,000 plus antibodies per second. It's pretty fast, right? They're terminally differentiated. They can't perform cell division. So it's the ones that proliferate, right? Some of them turn into plasma cells. Once you turn into a plasma cell, that's it. You're going to pump out antibodies like crazy, and then when your job is done, you're going to go through programmed cell death, apoptosis, and you're done. So we don't need you anymore, right? You've done your job. We don't want to feed you anymore. We don't want to house you. You must go because you're expensive. Right? And because we've got the memory cells, right? So if we see this again, we're just going to call up the memory cells, right? They're going to proliferate. Some of them will differentiate into plasma cells. And again, we'll get memory cells. So, so they're all produced from the proliferation differentiation of activated, whether it be naive, if we've never seen it before, right? Or memory B cells if we have seen it before. They only live about 2 days. Right? They work really hard for 2 days, right? And then once we've pretty much gotten rid of the invader, they're going to go through programmed cell death, apoptosis. So why do we call them plasma cells? Part of the reason is in that process of differentiation, they produce a huge amount of rough endoplasmic reticulum. You guys remember why it's called rough? What is it covered in? Ribosomes. What do ribosomes make? Proteins. What are antibodies? Proteins, right? So in order to produce that 2,000 plus per second, they need a lot of ribosomes to do it. And since this is a secreted protein, they're made in the rough ER, right? Then they'll travel to the Golgi apparatus, be processed, and sent out of the cell. So these cells, their cytoplasm gets enormous because their rough endoplasmic reticulum gets enormous. So hence the name plasma cells, right? They have huge cytoplasm. So we've mentioned this before, but we're going to home in on it a little bit more. So remember, each B cell produces its own unique antibody, right? And it can bind to a specific set of molecule configuration on an antigen. So say some large, maybe protein antigen. It binds to some little portion on that antigen, maybe 10 amino acids, right? That little section right there, it's recognizing. That little section where the antibody actually binds is referred to as the epitope, right, to that particular antigen. So antigens, especially proteins, are made up of, thousands of different amino acids arranged in different configurations. So you have differences throughout this molecule, and they're denoting that here by number 1, 2, 3, 4, and 5. These are all different configurations of amino acids that different antibodies are recognizing. Make sense? So a single antibody can only recognize a single epitope. So we're zooming in on it. You can see how they're different shapes to represent their different parts of that molecule that are unique. This antibody number 1 that's binding to epitope number 1 was made from, say, B cell clone line number 1. A totally different naive or memory B cell was activated and produced 2, 3, 4, or 5, right? So we have 5 different subsets, right, of B cells out there, whether it had been naive ones or memory cells that were activated, proliferated, turned into plasma cells and memory cells. The plasma cells started secreting their antibodies, right? So from that group 1, we get antibody number 1. From a different group, different B cell, completely different, you get 2. Completely different B cell line, 3. So when we respond to an antigen, because antigens can be quite complex, like you see here, that you get, say in this case, 5 different distinctly different regions on that antigen that we can make antibodies against. This is referred to as a polyclonal response. So poly meaning what? Many. Clones meaning those B cells clone themselves, right? But 5 different in this example were activated. So typically when you, especially a complex antigen like a virus or a bacteria, you're not going to produce just one type of antibody to one epitope. You're going to produce multiple antibodies to multiple different epitopes on that whole antigen. Does that make sense? We'll talk about next time. I'm going to skip over it today because I don't have time. We'll talk about monoclonal. So mono meaning 1. And so these are made in the lab, right? Your body, unless you're having some type of defect, isn't going to just produce one specific antibody.
So what does this do? At that point, what does it do?
What do you mean, what does it do?
What's its purpose?
Oh, what's the purpose? Well, think about this. Would you only want one antibody against an antigen? Wouldn't it be better to have multiple antibodies that recognize multiple sites on the antigen as opposed to just one? Right. And remember, it's random how our antibodies make themselves. So there's the possibility, hopefully, that again we're going to recognize multiple things.
So something like microphage will attach to it.
Yeah, oh, you're getting ready to what happens after an antibody binds to antigen. That's next time. You're getting ahead. That's the last thing we're going to talk about. All right, so I'm skipping over monoclonal and ELISA. I want to get to the structure of an antibody. So we're going to stick with the main class that's the most common. It's IgG. So these are proteins, right? But you actually make 2 different proteins and you make 2 copies of each. So what's denoted here, this big long stretch here, is one long protein, and this is an exact copy on the other side of that one big long protein. This one here is a shorter stretch of polypeptide or protein. And here's another shorter stretch that match. These guys are the same, right? The same amino acid sequence. They just made 2 copies. Okay, so each side of the antibody is kind of a mirror image of itself. Do you see that? Does that make sense to you guys? So we got one long protein and one short protein. So one of them is called the heavy chain. One of them is called the light chain. So which one is the light chain? A or B? A, because it's the smaller one, right? So therefore it was lighter when they were studying these proteins, when they separated them out. They're like, oh, one's smaller or lighter, right? So we call that one the light chain. So therefore, B I have labeled here is heavy chain. But these 2 are identical, the 2 heavy chains and the 2 light chains. So the 2 heavy chains attach to each other, as you see here, by disulfide bonds. The light chain and the heavy chain bind with each other by disulfide bonds right here. So when you look at this chart, right, the light green, do you notice, is our light chains, and the dark green represents our heavy chains. But within those chains, there's 2 distinct regions, what we call the variable region and the constant region. So variable means from antibody to antibody, is that the same? Variable, if it varies? No, it's different. So for each B cell, that's the part that's unique, right? So each B cell, so B cell number 1, its portion here that's, looks almost like candy cane to me, right? That has this alternating green and white stripes. This is the variable region. So both the heavy chain and the light chains have variable regions to them. But notice the solid colored sections, right, are what they say are constant, which means that that portion doesn't change much. And if you look, where are the bonds? Where are those disulfide bonds? So here's one, right? It's holding the heavy chain to the light chain, right? If the amino acid sequence in this area and this area wasn't such that disulfide bonds could form, would the heavy chain and the light chain bind with each other? No. And so you wouldn't get this structure, which is important to its function, right? And then look at, in the other constant region for the 2 heavy chains, that's how these guys hook up to each other. So if you weren't consistent in those regions, that binding wouldn't happen. You wouldn't get this 3-dimensional structure that we have. And of course, this is a stick figure, right? This is like drawing a person as a stick figure. This is an antibody as a stick figure. These are globular proteins that have an overall Y shape. All right, so we'll pick up here, and I'll have a few more slides for the outcomes that Lance really wants to get to, right? He wants to know, what happens when the antibody binds? Why is this good? Okay.